I'll just introduce our keynote presenters this morning, Joep de Ligt from ESR and Shaun Hendy from Te Pūnaha Matatini.
Kia ora.
Thanks, guys.
Tēnā koutou katoa, I mihi ana ki te Aotearoa, I mihi ana ki tēnei kaupapa o tātou.
Ko Joep tōku ingoa, ki te mahi o ki ESR.
Ko Shaun tākou ingoa, ki te mahi o ki Te Pūnaha Matatini.
He aha te mea nui. He tangata, he tangata, he tangata. He putaio, he tangata. Thank you very much for having us. As you just saw in the mihi, we're going to do a bit of a duo act here for you today. And we'll be talking about the COVID-19 response. And as Minister Verrill very eloquently pointed out, as New Zealand, Aotearoa, we managed to pull off something quite special. And that's what we're here talking about. But before we do, I just want to briefly flash back to 1 year ago when my colleague Richard Dean was doing a keynote at eResearch. And he was talking about public health investigations, one of the first ones being in London, where a water pump was identified as the source of an outbreak. And he was saying that he would love to see more digital technologies brought to bear on epidemiological investigations. Little did he know. So one of the things that Richard was talking about was training Pokemon. And since this year's theme is leveling up, I thought there was a nice analogy there. But of course, there's only one Pokemon that can help you with investigations of a pandemic, and that is Detective Pikachu. So in good ESR style, I will put on a slightly silly hat, and we will get proceedings started by setting the scene of why these two guys are here on the National Women in Science STEM Day. That is because our respective teams pulled off something quite unique. And this is a quote from the World Health Organization that was used in an article that was published in November. New Zealand uses science to avoid coronavirus lockdown. We're keeping safe without locking down. How do we do that? That's through a unique combination of targeted modeling and rapid genome sequencing. And that is why we are here today, because that's the two topics that we've been involved in. And there was another thing that I took away from yesterday morning keynote, which was a lovely beginning to end plot. And I thought I have to use that. So that plot is New Zealand's Cinderella story. What you see on the left-hand side was our preparedness score before the pandemic, which was good, but not great. And on the right-hand side, you can see where is the best place to be during the COVID pandemic, where we are number one, which was pretty cool to see. But as you can see, I've drawn the Cinderella line to sort of not quite at the end of the axis, because we're not out of this yet. So with that, I'll hand over to Shaun about what our plan was.
Yeah, look, we're going to be jumping back and forth between the two of us over the course of this morning, which is actually how our science has worked. We have been working alongside each other and exchanging information, and at some point, sometimes the modelling is the key thing, and other times the genomics has been the key thing. But actually, it's the combination of those two that's really powerful. So we didn't come into the COVID-19 pandemic without a plan. There was a plan that was sitting on the shelf at the Ministry of Health, the New Zealand Influenza Pandemic Plan. It was developed in 2017. Our team actually had a little bit of input into this. We were, which I'll talk about in a minute. It basically, if you go look at this plan, it doesn't describe what we did, right? If you read through it, it describes what we'd now call, right, what's very familiar language to many of us, as a flatten the curve strategy, right? That's we're going to attempt to control the disease spread to the extent that we can keep the number of cases that are going to be hospitalised within the capacity of our healthcare system. So that was the plan. That's that's what we had developed. And there were a number of phases, and you may remember the ministry and government ministers talking about those phases very early on. But that rapidly disappears. Some of the things that are good about the plan is, it does make the point that decision making will have to be rapid, right? The plan relies on you making good decisions early on and implementing those things rapidly. One of the things that wasn't quite so good in the plan was it does not discuss how you bring new science to bear. Okay, it's complete vacuum in this plan. If there's new science that's needed, either because it's being developed and has the potential to make an impact, or because of the particular pathogen that you're dealing with, right? This plan doesn't tell you how to go about integrating it, and so although it acknowledges uncertainty, it doesn't really talk about uncertainty in the science or how you bring new science to bear. So, and of course, there was new science that was needed, and COVID-19, the novel coronavirus that we were dealing with last year and this year, does give us challenges that weren't anticipated in the plan. I mean, it's interesting to just reflect on why. One of the key things that makes this such a difficult pathogen to deal with is that there's significant transmission by people who either have very mild symptoms or are pre-symptomatic. So in fact, the viral load builds up and is actually peaking prior to, and many people prior to you actually developing symptoms that tells you that you have the virus. And that means that relying on people to, based on symptoms, to go get a test and to self-isolate is not necessarily going to contain the virus. Whereas something like the original SARS virus from 2 decades ago, people developed symptoms prior to peaking in infectiousness, right? So that made it a much easier virus to deal with from a public health perspective. This one is that we're dealing with at the moment has this difficulty that you can't tell who's. It's not easy to tell who's infectious until they develop symptoms, and so that's a key difficulty. So, it's not as though we were all sitting around completely satisfied with that 2016 plan. In the science community, there was work being done. Te Pūnaha Matatini had gotten involved in 2016 in a project to look at seasonal flu. And the idea was, could we combine the genomics with some modelling and maybe some interesting data sets that tell us about the mobility of people? And so it turned out that we could. We were able to look at seasonal flu, and we were able to show that using some very coarse-grained cell phone mobility data, right? So this is just literally how many movements from DHB to DHB take place every day. That we could actually we could do a pretty good job of predicting how a seasonal influenza epidemic unfolds in New Zealand. So that was back in 2016. We talked to the Ministry of Health about it when they were developing their pandemic plan, and they said, well, that's great, and then we never heard from them again until last year. There was preparation being done. There was work out there that we knew could be used in the event of a pandemic, but it really hadn't been integrated into the plan or really thought about deeply.
So what we'll be talking about is how does two different things that were already ongoing on a sort of best effort basis by the scientists, how that was accelerated quite rapidly over the last year. So how did we level up our science to the COVID-19 response? So on the modeling side, Shaun and the team really did a lot of work on making these New Zealand-specific models because our populations are not like other populations. There's specific dynamics in between movement and all those things like age makeup and all the other things. There was a better integration ongoing with the epidemiological data. Shaun just mentioned the telephone data, movement data. How can you integrate those kind of things? And also, how do you integrate that into the decision-making process? How do we make these new scientific pieces of evidence make them get to the decision maker so that they can make informed decisions. On the genomics side, historically sequencing happened from isolates. So that's where you take the pathogen out of the sample and grow it up in the lab, and then you can start do genome sequencing. We were moving on different projects, we were already moving to do that directly from clinical samples because that saves valuable time. And that's that rapid turnaround. So other than just using that as clinical samples, we also needed technologies that were going to give us answers sooner. And a recurring theme, also better integration with the epidemiological data, so that you can get that rich insight from these different data sources combined into one.
So yeah, where were we at the start of March? So as I've said, we kind of had, we'd done a little bit of work back in 2016. We'd actually continued as a research centre to sort of tinker with infectious disease modelling. It was not our core activity, but we've been doing a little bit of work in those intervening years. At the start of March, you know, what did New Zealand have? Well, we had access to some very, very simple deterministic models for COVID-19. So actually the very first simulations that were done for a New Zealand context, I've got the example here. This is Nick Wilson, who's at the Otago School of Public Health here in Wellington, and you'll recognise a few names there, including Aotearoa, a new minister, and Michael Baker. And they were using some code that was actually developed in Germany, and it's not New Zealand specific, right? And you can see their preprint. It's a hypothetical European country, and that, you know, they're basically treating New Zealand as if it's a small European country in that modelling, and that was really where we were in March. We'd done something very similar. I'd coded up something on a Sunday night in early March, and so at TPM we had a very simple model as well. And these are kind of these models. You know, there's a lot of shortcomings of these types of models, but they give you a sense of what kind of long-term outcomes you might be looking at. But very quickly, New Zealand moved away from facing these widespread outbreaks. Right, we moved into lockdown with the goal of eliminating the virus. Those models, those very simple models, simply can't be used to look at elimination. You can't use them to do short-term forecasting. And so the types of models that we had access to in early March, you know, simply weren't going to be fit for purpose. So we actually did need to crash start a modelling program in New Zealand to really develop the kind of tools that we were going to need over the next year. And so this was kind of this is how things evolved in Te Pūnaha Matatini. We started off working with these deterministic models. They're known as compartment models. I've got the jargon, which is SEIR. Don't worry about that. You know, they're good for giving you a sense of the long-term outcomes, right? But that wasn't what we were needing to look at in New Zealand. But you know, you can run them on a laptop in a few seconds, right? They're really simple. You can play with parameters. You can do sensitivity analysis very, very easily with them. But we knew they were going to be insufficient. So by mid-March, we were working on stochastic models. So these are models that try to get to grips with the stochastic variability of the disease spread, right? You know, and you remember from think back to March, the daily case counts that we were getting, right? It wasn't following a nice smooth curve, right? We were getting jumps up and down, and you know, you get a high number one day and a low number the next day, and you'd be wondering, well, what did that mean? Were the numbers coming down? So we were using a stochastic model to try and analyse that. These models you have to run ensembles, right? So you don't just run it once. You might run it. I mean, sometimes we run these models 1 million times to get a sense of the variability we're looking at. But you can sort of run those things on a laptop in an hour, which turns out to be a useful timescale for responding to prime ministers. But there are also shortcomings of those models. You know, you can't look at single interventions, right? So with that kind of model, it's very difficult for us to say, well, what would be the effect of closing a school or shutting down a particular region and keeping particular types of businesses operating. So we were working at the same time on what are known as network models. Right? They're computationally much more challenging, and so we really need a day with a lot of compute nodes on NESI to run these, and we might be looking at ensembles of about 100 simulations with these. Right? So they're very, very detailed. There are 5 million individuals in these simulations, and we can see, spread through workplaces or particular schools or places of residence. So that was kind of the timing of the tools that we built over the first 6 months last year. And of course, it turned out that by late April and early May, right, we needed to have these stochastic models. Right, we needed to be giving advice to decision makers. In order to, for them to have some confidence that stepping down alert levels was the right things to do. So we were providing plots like this. This is basically a given date, so this is late May, and we're providing these graphs to the cabinet to give them a sense of, if they step down alert levels now, what's the chances that we've eliminated the virus and we won't have to go back into lockdown? We had to do these very rapidly. Back in May, we were in this weekly cycle, but at other times we've been in daily cycles where we were having to turn these models around, and so we had to develop a workflow that could deal with that. Right? Obviously, sending your results away to a journal for publication to appear, 3 to 6 months later. It's not going to be timely enough for Cabinet. They're going to get a bit annoyed and wonder what they're paying for.
So the other side of the story that we're talking about was the genomic side. And the question was really, so where do we want to be? For us, it was about a year ago, we were seeing the reports coming out of China, and countries were starting to lock down. And we were like, where do we want to be with our genomics response to respond to this virus? And just like Shaun, we were like, we need to throw the proverbial kitchen sink at this thing. We need every piece of data that we can get. So we said, we're going to sequence every single case in New Zealand. And that was not part of the game plan either, but we still tried. So how did we go about trying to do that? Well, there was a really awesome paper in Nature a couple of years ago about genomics-informed pathogen surveillance. And it's really about how do you take these technologies to where they matter and inform decision-making. So it was successfully used in both the Zika and the Ebola outbreaks, which are arguably quite remote and under-resourced areas, where they were able to take these genomics technologies into the field and put it into the hands of the people that were actually working on these viruses. So we said, well, I think that's the kind of technology that we need to bring to bear on this. It needs to be rapid, it needs to be deployable. We don't know what we're getting ourselves into. So how does that work? Genomic Surveillance 101. We have a lot of armchair critics in the audience, so I'll keep it simple. But it basically, you have an isolate. In this case, those were these nasopharyngeal swabs that people were kindly getting to test if they had the virus or not. But we can then also use that to sequence. In this case, we sequenced the RNA, not the DNA. We mapped that to the reference genome. So thanks to China, we had a good reference genome for this particular virus. And we can then look at differences in that sequence. And those differences, they are basically the history of the virus, where if you look at the graph at the left-hand side with the people, where someone has developed that orange C mutation, if they infect someone subsequently, they will carry that mutation as well. And that chain then continues, and then we can make what we call these phylogenetic trees, which is reconstructing the evolution of the virus and how it has mutated. So what are the kinds of questions that we can ask if we use genomic surveillance? Well, we thought that we could assist outbreak investigations because it's an independent and additional line of evidence. Viruses have a tendency to not forget their mutations, while humans might forget where they've been, especially after a night of drinking. You can estimate the number of introductions into the country, where and how often is it coming into our country, where is it coming from, how transmissible it is, something that Shaun just touched upon, the R0 value. In the early days, we didn't know a whole lot about the virus. There were some rough indications about how many people you would infect if you had the virus. But genomics provides a way to track that. And we thought that we could investigate transmission chains. So in these communities, how is it spreading within that? Because you can link the chain of transmission of whom went to whom to whom. So this was a pandemic in a new era. It was a pandemic in a new era of genomics and in Twitter. So on the 31st of December, the WHO was notified of the virus, and on the 12th of January, we had a genome. That is earth-shatteringly quick. That tweet by Eddie Holmes will go down in history, and if he ever gets the Nobel Prize, I hope he wears it on his shirt. Because on January 11th, he already posted about that they were making that available for the world to use. That meant that people could start making diagnostic assays to test if you had the virus or not. And it also meant that people that were making smart RNA vaccines could start right then and there, right? And on 23rd of January, we had the first protocol to sequence the sample directly. So to take a clinical sample and to get a genome out of it. And that was made by the Arctic people in the Arctic Consortium. And there's a tweet from Josh Quick saying, we have the reagents. You don't need to go through difficult ordering things. Just put up your hand and we will send you so that you can test this protocol and that we can see if it works. So we put up our hand. And on the 28th of January, a shipment of those reagents was on its way to New Zealand, just up the road here. So we were getting ready to take this thing head on, and I think we managed. Now, one of the things that a lot of people ask, as I put a slide in about it, so where did the virus come from? This is another thing where genomics was really useful. Since that there was some surveillance going on of wildlife in China. And what you can see here in the graph is the samples in green were derived from pangolin, and the sample in gray was from the horseshoe bat. And the sample in red is the first sort of genome from the coronavirus. And its closest ancestor is in bats, and just yesterday there was another publication that they found another bat sample that is very close to that, but there's probably an intermediate host since that the most common ancestor is placed about 30 years back, which means that there has been ongoing evolution over that time scale where it could have been in other animals before it jumped to humans. So there's another one of those things where with genomics they can now rapidly identify which areas they need to be looking for that human-animal interface where the jump happened, and hopefully we'll find it. So can genomics inform decision-making differently? Because historically, people here in New Zealand might remember the Havelock North story. It is retrospective. It helped understand that outbreak and it helped identify the source of it, but that was typically a matter of days or weeks, not hours. And as we just heard from Shaun, ministers and PMs, they want hours, not days or weeks. So what changed from going from retrospective to real time? So this is the protocol that we use on a very high level. Why can we do this with this particular virus, and why are we not doing it for all pathogens? That's a question, a very good question. It's a stable virus. It doesn't seem to recombine a lot. So the order of nucleotides within the virus is stable, so it doesn't jump around, because that will mess with your primers real quick. It has a low mutation rate, so it's not mutating away from your primers, so you can use the same primers over a period of time to capture it. And it's a fairly small genome. It's about only 30,000 bases. So that means that you don't need too many amplicons to capture the virus. So how does it work? Very quickly, it's an RNA virus, so we convert it to cDNA because that's a lot more stable to work with. We amplify the virus specifically through these PCR amplicons. We make a library of those amplicons, and then we sequence and analyze in real time. Easy as. So actually, this is the technology that we use, and there's been a lot of foreshadowing from Miles's talk already, and I'm just going to add to it. If you want to know more about that technology, go talk to Miles, but it's basically, it's a type of sequencing where you can analyze the data in real time. So what you see there is a molecule going through a nanopore, and there's a electrical signal being detected based on the type of sequence that is flowing through that pore. And it is that signal that we use to identify the different nucleotides that are present in the virus. That means that after we start sequencing, and that because this thing is sequencing at over 300 bases a second, we can start analyzing the data after an hour, because we will have read the whole genome multiple times, right? That's something you can't do with other technologies, and that's why we chose this one. The other thing that's on this slide is the rapid barcoding protocol that was developed by Nikki Fried and Olin Solander and their team at Massey University. And they actually made a slightly different set of amplicons than the one that we originally used back in March, which has less of them, and they highly optimized the protocol to do that. And their protocol is still a protocol that we use to get these really rapid cases done. And that means that from receiving a sample to looking at the data, in emergency scenarios, we've been able to do that within 8 hours, right? That's from a sample to a report that can go to government to inform the decisions. This is the actual data for those that like to see that kind of thing. I'm one of them. So on top, you have an overview of the pieces of amplicons that we use to target the virus, and we can see in real time how they're performing, if there's bits of the genome that we're missing, or if we're getting a nice even coverage. And then below that is the actual read data, where you can see that those amplicons, they overlap slightly, so that the region where we capture it is sequenced in another amplicon, so that we can see if there's mutations that are messing with our protocol. And you can also see that we sequence quite deeply, because we do want to be sure that we pick up the right mutations. So why nanopore sequencing? James put it quite eloquently. A lot of people wonder, like nanopore has slightly more errors. It's not a lot. So can you do that? Yes, you can. And you can do it in hours rather than in days. And they actually had the time and effort to write a nice paper about it, so that we can just reference that when people ask that question. But as Shaun mentioned, we were changing a system under pressure. Decision makers were getting information from left, right, and center. People were locking down, staying at home. There was chaos in the system. And the plan that was there was not necessarily well prepared to take these information in.
Yeah, so one of the very first things we had to do was to try and figure out how we're going to get this information to policymakers in the right way. We did want to do peer review. We couldn't obviously do traditional peer review. So early on we established an internal peer review team who were just basically independent of the work, but could look at the work, give us rapid turnaround, rapid peer review comments, and that we could incorporate that back into the work. We also had a policy review process, so we worked really closely with about two or three policy people, some in health, some in Department of Prime Minister and Cabinet, and they could look at what we were doing and tell us whether, you haven't quite answered the question that we asked, or that's great, can you then look at that? And so both those things were sort of rapid turnaround processes that you might be doing overnight to quickly get results out. Quite early on, we also realised we were creating some confusion, and that's because there were kind of two users of the work. One was a sort of at the operational arm of the health response. So this is people wanting to know how many tests are we going to have to run in Northland this week, right? Because they need to get set up testing centres, they need to get supplies for the tests, etc. And versus people who are going, what would happen if we change alert levels next week, right? And so we realised early on we had to separate those things out. Because we couldn't send the operational people hypotheticals about what might happen with alert levels, right? And so we set up a process where we separated those two things out. So we'd have scenario sandpits where policymakers could ask us to try this, try that, and we separated that out from the operational side of things, where we had what we called working scenarios. So we had agreed sets of we will move to level 2 at this point, and you can base your decisions around that. And then really important for us was the public release of what we were doing. And so again, we tried to within a week or two have the material that we'd supplied to makers up on our website, and that was to provide transparency. I mean, it's also pragmatic, because of course people are putting in Official Information Act requests for your work, so if you get it out rapidly, then you can just point them, rather than having to deal with an OIA, you can just point them at the work you've released. But also it meant that the public could interact with and understand the science on which people were making decisions, or which governments were making decisions. We had to get data integrated into what we were doing, and I have to say, early on, we got our data by watching Ashley Bloomfield at the 1 p.m. stand-ups, right? So we'd all turn on our TVs and watch and write down what he was saying, or getting it off the MOH website. So early on, there wasn't a flow of data in. What we needed? Well, we needed patient-level data for the stochastic model, right? Because we were actually seeding that stochastic model with the real cases. So if we didn't know when someone had got infected, right, then we were having to make guesses about that in our stochastic model, which might actually change the outcome. So we really needed that. And then for our network model, we're actually using the integrated data infrastructure, which is StatsNZ's linked administrative data. So it has information that links, say, households to schools, to employers. Now, we didn't pull that literally out. We pulled out a pseudo representation of that. So it's a representation that has the same network structure as New Zealand, but it's not the actual structure of New Zealand. And so we needed to put those things in order to make these models work effectively. And here's kind of the theory of how it should have worked. We have a public health unit, so they're working directly with patients, with people who've been infected, right? And so they're the agencies that actually carry out the contact tracing. Their information, in theory, should be fed in to EpiServe, which is this database maintained by Ministry of Health. Some information goes out to ESR, and then ESR, of course, which is doing managing testing and so forth, information goes back into EpiServe. That's kind of owned by Ministry of Health, right? So it should all flow into Ministry of Health. Stats New Zealand was assigned to work with us to be our interface between health and Te Pūnaha Matatini, and so this all should have flowed really nicely. Instead, we had to send boxes of chocolates from time to time to ESR in return for them running search queries and downloading them for us and getting them by email. So, a lot of, there was a kind of a nice theory about how it should work, but especially during that first lockdown period, the systems just weren't there, right? The PHUs were running different systems, some of them still paper-based. Ministry of Health wasn't really an operational organisation, it was very much a policy shop, and so they really weren't used to doing things like this at speed. And so, the box of chocolates was the solution to that problem. Now, actually, things resemble the ideal diagram. There's still the odd, piece of chocolate sent around the country, but mostly things flow as they should.
So one of the other things that the system was not used to was that we made a request for all positive samples to be sent to us for genome sequencing. That's not something that people were used to. So as you can see here in the graph, over time we got better at convincing people that it was a good idea to send those things as soon as possible rather than keeping them in your fridge for 180 days. Now actually all samples are being referred directly. So over time you can see that band is getting a lot narrower and a lot better. So there is still a fraction of samples that we never received, and that's just because of the chaos that the systems were in and that this was not planned for. They got either lost or they were destroyed before actually people referred them to us, or because of quality concerns around those samples. But we still got a very good representation, so we're only just behind Australia in terms of the numbers of cases that we have been able to sequence, and that has allowed us to make really well-informed decisions based on those genomes.
So now we'll have a little bit of a scientific interlude now, just to sort of throw in, what are some of the things that we learnt, some of the things that we did with the models and the genomics. One of the things we're able to do with the EpiServe data is we could try and reconstruct the clusters that we were seeing. Of course, people were talking about these during the MOH briefings, but there was no guarantee that we had everybody in those clusters, and so using Monte Carlo techniques, we're able to reconstruct some of those clusters. One of the things we found, and of course you might have heard of this from overseas or through the genomics work, is that this is a disease that spreads via superspreading. So about 20% of adult cases were responsible for 65 to 85% of the transmission. So there's a small number of cases that are generating most of the transmission. Only of the detected cases, only about 2% ended up being asymptomatic throughout the course of the infection. If you look overseas, overseas numbers suggest that number might be higher. So that's possibly an indication that we did miss asymptomatic cases, which shouldn't be surprising because, of course, the way that the testing was being prioritised, we were asking people with symptoms to go get tested. So if it wasn't obvious to people that they should go get tested, then they wouldn't necessarily have. But those cases they were less infectious, and that's consistent with overseas. We were able to look at, we talked about, I didn't actually go much into that number R0, right? We're probably all familiar with that number R0 now. That's something that we discussed at length early on in the outbreak. We're able to look at the daily R0, which is really, it's quite an interesting quantity for understanding the progression of the March-April outbreak and, of course, the August outbreak. And I've just split it into two cases here. We've got imported cases, so these are cases that have come in from overseas, versus domestic cases. So these are people who caught the disease within New Zealand. And interestingly, what the important thing is that R0 value above 1 or not, right? If it's above 1, then your epidemic is growing. If it's below 1, then your epidemic is declining. And you'll see, in mid-March, so that's that weekend when we were aware of it, but still going about going to weddings and conferences, right? That number was something like 3 or 4, and then you can see, as we became more aware, it declined to something like 1.5. So in the weeks leading up to lockdown, right, we'd stopped partying. We were skipping the bar, and we'd reduced that very large R0 value, but the epidemic was still growing. We went into lockdown and it dropped below 1, right? And so that's when we started to eliminate the virus. You see these interesting spikes here late. Luckily these spikes were occurring when we had very small numbers, so they didn't lead to substantial spread. But these were the long weekends. So this is the Anzac Day long weekend, and this is Easter. Okay, and that spread was taking place in healthcare facilities, so it probably represents something that was taking place in the way staff were being rostered. And really interestingly, R never got above 1 for the imported cases, right? It was below 1, but nonetheless we had enough of them that they were able to seed extensive outbreaks.
Yeah, so from a genomics perspective, we looked at the same sort of data, and one of the things that we saw, as we touched upon, is that some DHBs really got what we were asking for, and they were able to send almost all of their samples, but there's also some regions that we didn't sample quite well in that first wave, and that's luckily improved a lot, but it is something in terms of preparing the system that's interesting to know. We estimate that there must have been at least 277 separate introductions, so that's what Shaun was referring to, that the R0 might not be high, but there were a lot of them that seeded the epidemic here. Luckily, thanks to the lockdown, of those introductions, only about 24% led to other secondary infections, and only 19% led to ongoing transmission. So that's where that lockdown really helped to stem that tide of these incoming travelers seeding that. As Shaun was referring to, some of those super spreading events had very, very high R0s. So our largest cluster at that time was the wedding event, where based on the genomes and the epidemiological data, we estimate that before lockdown, the R0 was about 7. So that's a big super spreading event. But that with the lockdown, for that particular cluster, that dropped to below 0.5. So there's only those small households afterwards that got infected, but nothing major onwards from that. So this is really good evidence from different sites that these lockdowns were really needed and that they were really effective.
So coming out of lockdown, I guess everybody was, it was a big relief coming out of lockdown, including for us working. Life went somewhat back to normal. I think there was a little bit of disappointment that we hadn't quite managed to bring the genomics and the modelling together in the way that we kind of anticipated a few years ago. Because we were busy trying to build these systems, they hadn't quite come together. But it turns out that there was going to be another opportunity for that. So over the June-July period, we didn't stop working. We were continuing to improve our models, and in particular, the Department of Prime Minister and Cabinet asked us to adapt our model for reincursion scenarios. Okay, so this is where, as we're all familiar with now, we have a single seed introduction from an MIQ case, for example, and so we had built that by the end of July, and we were going to actually have a little trial run sometime in August to just see how that modelling went. In the meantime, the genomics side of things had also really stepped up its game. It now become a routine part of reporting, right? And it was now being integrated into the epidemiological data in real time. So, on the night of August 11th, I was actually at the pub about 6 p.m. I'd had to give a big presentation at the university that day, and so I was having a beer to relax afterwards. I noticed an email pop up on my phone, with urgent, and the body of the email just said, that thing we asked you to do in July, right? That's now become much more urgent, right? And after taking a phone call, we found out that there was a symptomatic individual in South Auckland. They'd been symptomatic for 5 days, which means they'd been infectious for quite a long period of time. And there was no known connections to frontline border workers. I've got a picture of my colleague Alex James. There were kind of three of us who were kind of on the core team for the modelling, who would do the rapid response. The other person was Michael Plank. He was at the basketball that night, had his phone turned off, so we couldn't get hold of him. Alex, I was at the pub. I did have my laptop, which was a start. Alex was at home wrangling 2 small children through dinner time. But between Alex and me, back and forth, and we got a report, right? And the question we had to answer for decision makers was: we've found this one case. How many other cases are out there? Right? And so in our modelling, right, there's kind of 2 types of scenarios that we look at. One is if someone was connected to the border, right? So if you're a step or 2 from the border, right, then that and it came in through the border, then that tells you that the outbreak will tend to be smaller, right? Because you've caught it earlier. And so if there was a connection to the border, then we were saying, okay, there's probably between 0 and 22 cases out there, with an expected number of about 14. In that situation, decision makers may quite possibly have trusted our contact tracing service to keep a lid on the cluster, and we might not have had a lockdown, versus the other scenario, if there was no connection to the border, then we're talking about around about 40, maybe 50 cases, and that's starting to get at the point where you'd be really concerned about the Auckland public health units being able to deal with that outbreak. So the thing was, well, how could we tell whether this was connected to the border? This was absolutely crucial for understanding whether we should go into lockdown or not.
So yeah, luckily by that time, Genomics had gone real time, both in the sequencing as well as in the reporting. The first couple of cases we were able to sequence within 12 hours, and we saw that it was lineage B111, a lineage that will go down in infamy. It's been renamed since, so we can forget it. But it hadn't been seen in New Zealand since March, and this is where it was really important that we did have a good representation. And especially in Auckland, we had about 80% of the samples that we had received, so we were fairly certain that we had a good representation of the types of viruses that had been doing the rounds in Auckland and across the rest of New Zealand. So the fact that it hadn't been seen since then meant that it was unlikely to be linked to one of these undetected community transmissions. But we also didn't see a link to managed quarantine. Right? So this is one of the scenarios where there's a lot of unlikely events, but one of them happened. And we're discussing over dinner yesterday, and there was a nice analogy with farmed salmon, where if you catch a salmon in New Zealand, just somewhere off the coast, where did it come from? It's not naturally a naturally occurring fish here, but there is farms around somewhere, so there's unlikely probabilities, but one of them must have been true. When we sequenced more cases, there was increasing evidence that this was one transmission chain. They all shared one mutation that was never seen before globally, and that we also have never seen since because we eradicated it. So that's the figure on the top there. That's the global tree of all the BB1 sequences, where in red is the New Zealand cluster. And you can see that all of them group to that one single node phylogenetically, and that there's nothing else internationally there. The other thing that we were able to do was there was that one staff member that tested positive that was actually not linked to the cluster but to another traveler. And there was that one case that was infected in the quarantine facility where we were able to pinpoint one of these two individuals must have been it based on the genomes. And then the epidemiological data came in where they said, well, they only had contact with that one person. So we immediately knew that that person must have been infected by that person, allowing them to investigate what breach of PPE or protective measures might have occurred or how that could have happened within that very secure environment. There were some other things that we did in terms of the investigations. Since Auckland, we've had closed cases for every single one, which I'm still quite fond of. So for every single incursion since then, we have been able to link it directly to a border worker or an MIQ facility. So there was the fisheries workers that we had, where there were two nurses that got infected and they were linked to different clusters within that. There was the person that went onto a boat that was infected by one of the workers that was brought in from abroad. That was a good collaboration with the Australians because the person that carried that virus onto the boat was actually on international waters by the time that we figured out that we had community cases. But luckily our Australian counterparts could pick them up on the boat and swab them and confirm that indeed they were the source of introduction for that cluster. We did investigations of in-flight transmission. It's one of the most comprehensive pieces in terms of knowing that it was really in-flight transmission on one of those long-haul flights into New Zealand. And more recently, these variants of concern, where there's these variants that look scary or dangerous. And by doing real-time genomics, we can keep tabs of how this virus is mutating and which ones might be more infectious or which one might be escaping immune systems, or hopefully soon there will be vaccines, but there might be particular versions that escape that vaccine. So by doing genomics, we can keep track of which ones that are so that people can update their vaccines to act accordingly. And as Shaun was referring to, all this was happening in real time, where you are making a report that the next morning will be televised on national television, where then there's a whole lot of questions that come your way, these information requests, and all those other things. And I think the important message for us that communication was and will be key. So here are two of the visualizations that were made by Suksi and Toby for a much wider audience, where they're explaining the modeling and they're explaining the genomics, so that we can get on with our jobs. So there's one thing that was also happened, and that's quite rare in science, and especially in pandemic responses, where you can say, this is where our data made a difference. And that came in November, where there was, again, a lot of worry about these community cases, and do we need to lock down? As we've seen in Australia, once they get a community case, they will lock down, right? That's a crude measure in the grand scheme of things, but it's a very effective measure. So they're not taking any chances. But we had the luxury of having both of these data streams feeding in real time, and they said, we're not going to lock down. And in hindsight, that was the right call. But it was based on the evidence. It was based on the evidence of the modeling and of the genomics, that we knew that all of this was linked. We knew that there were no further contacts that were missing. We had a good contact tracing system. And the modeling showed that it was likely to be contained. So if we assumed a 3-day lockdown of Auckland, that would have cost Aotearoa over $100 million. This was made possible by an science investment of just over $2 million, right? So I don't want to make fun of the housing crisis, but that's better return of value. So what's next? We'll leave you with some of those thoughts and hopefully spark some discussion. There's ongoing modeling of the vaccine rollout strategy, so that's why our Cinderella story is not quite done yet. We're still dancing at the gala. We're still having our glass shoes on, but they might shatter. Right? How are we going to roll out these vaccines? How are we going to make sure that they are effective and that they protect the right people, and that we roll that out in an equitable manner? We will be monitoring if there are viruses either internationally or locally that escape this vaccine, so that we can adapt to that. And then our shared dream is that this will feed into a long-term preparedness for these types of events, so that that Cinderella line can keep going plus and has a nice, stable, utopian, lovely world. It takes a team of 5 million. There were a lot of people involved, a lot of institutes involved. I think this is still a testament to New Zealand science collaborating, something that we heard the minister talking about. All of the different universities and institutes, were so many people that reached out to us. Do you need a hand? Do you need reagents? Do you need anything? Let us know. And that is just a very warm, fuzzy feeling that I hope that we can continue, because everyone doing their part makes for a very good place to live. So while we roll our credits, happy to take questions.